Okay guys, now we're going to hit biochemistry. 51 questions on your test. You know this is one of your big 5, so we got to dominate this. If you're starting this video and you're thinking, man, I can't stand biochem, hang with me. There was a time when I did not like this section either. I remember as a student just feeling like, oh, biochem. But now I really enjoy it, and especially I like it after looking at the old released exams for the NBDE Part 1 because we see a lot of consistency in what they're asking. To be honest though, there were a lot of random kind of challenging questions that they would ask. And so I've gone through those obviously, and I'm going to point those out. So every once in a while, every couple pages, I'm going to point out one of these random and challenging ones. And you're probably going to roll your eyes and think, geez, that was hard. But don't worry, after you make a note of it in your book and you come back and just hit it 1 or 2 other times before the test, you'll be fine. So I just want to kind of prepare you for that. Just roll your eyes with me and make a note of it, and we'll get through it together, okay? So as we look at this, one of the kind of the framework on how you want to think about this chapter is this, okay? You're going to have, as the general overview, proteins, lipids, carbohydrates, and nutrition, okay? That's the way we want to think about this. So we're just going to nail those 4 areas, really hone in on nutrition at the end, okay? That's going to be important, and I think I have some ways that really help make that easy. So as we start off in this chapter, we start off looking at cell structure. Let's just take a glance down at your notes. Cell structure, cell communication, and signal transduction. That's like the first 3 or 4 pages. Those first 2, cell structure and cell communication, are covered already in histology. Let me show you this visual though up on the screen just to kind of remind you of some of those key points I want you to know about the organelles, because I want to make sure that you have these points down. So take a look at that, and then we'll get right into signal transduction. Okay guys, so take a look at signal transduction with me in your notes, okay? And remember, the whole point of this is to create a cascade that alters gene expression within the nucleus, okay? And as you look at that paragraph, scroll down to where it says CAMP in bold, okay? Now look at the sentence right above CAMP. It says adenylyl cyclase is an enzyme that produces multiple secondary messengers known as CAMP, right? So anywhere in the book where you see adenylyl cyclase, correlate that to CAMP. So if you increase adenylyl cyclase, you will increase CAMP. If you decrease adenylyl cyclase, you will decrease CAMP. Now, just to show you the practical application of that, if you haven't watched the general physiology video yet, within the first few pages of that, you'll see alpha-1, alpha-2, beta-1, beta-2 receptors. You'll see their mechanisms of action, and you really want to hone in on alpha-2, beta-1, and beta-2, because those 3 receptors, just make a note of that, and take a look at this visual here. All 3 of those receptors, alpha-2, beta-1, beta-2, are going to alter adenylyl cyclase and therefore alter CAMP, okay? So that's just a quick tie-in on how they've asked that in the past, so pay attention to that as you're looking at it, okay? Pay attention to that. Now, as we get in to the next topic of chemical bonds, look at that in your notes. You're going to see covalent bonds right there in bold involve the sharing of electrons, right? So they're going to be the strongest of all bonds. You're going to see in a second that amino acids are bound by covalent bonds, okay? So pay attention to that. You're going to see non-covalent are listed right there: electrostatic, hydrogen, and van der Waals. Those 3 are examples of non-covalent, and that's what they wanted you to know on an old test. I wouldn't study those in depth. Obviously, they could ask you a picky question about it, but in general, they wanted you to know they're not sharing electrons, they're weaker, and they're examples of non-covalent binding, okay? So that's the key things, just some quick points there. Now let's get into proteins together, all right? Okay guys, to help organize proteins for you, for this first video here you're looking at within biochem, I want you to kind of organize it this way in your mind, all right? And these are going to pop up so you can see them here. Number 1, I want you to pay attention to the fact that proteins have nitrogen. We'll get into that in a second. Number 2, I want you to know tyrosine really well. Number 3, I want you to pay attention to antibodies, and we're going to talk about that in a minute, particularly the functions of the different antibodies. Number 4 is to know collagen inside and out, okay? That's really important. And number 5 is amyloblastin, all right? So those are going to be the 5 kind of hot topics I'm going to hit over the next 20 to 25 minutes. So when I get to those, really hone in on those, but I want to give some outline to the lecture as we get rocking and rolling here together. Okay, so let's look down at the notes together underneath proteins. Look at protein structure. First line, it talks about proteins are comprised of amino acids linked, what? Covalently, right? What does that mean? Shared electrons. By what? peptide bonds. They could also mention the term amide binds, right? Same thing as a peptide bond, shared electrons, right? It goes on here to talk about the 20 different types of amino acids, and you can see them here listed for you. There's a few I want to kind of jump off the page at you, okay? Number one is aspartate. This isn't a hot one, but it has been mentioned a few times, and the one thing they want you to know about aspartate is shown right here on the screen. Take a look at this. The reason why we want to stress that is anytime, guys, you see something about a ketone body, pause, okay? Because ketone bodies have to do with ketoacidosis, type 1 diabetics. So anything to do with ketone bodies, we want to know it. So that's why you should know that point, all right? Now, if you go on to tyrosine, that's going to be another one you're going to see on the next page. Tyrosine has a precursor. What makes tyrosine? Do you guys remember what that is? That's going to be phenylalanine, yes. So make a note next to tyrosine. Phenylalanine is what derives tyrosine. And then you're going to see once we get to tyrosine, tyrosine can make several other things, okay? So take a look right here at this visual. I want to show you what tyrosine can form, because these also have been shown on past tests. The next thing I want to point out is the amino acid structure. So you can see this in your book, but also take a look at this visual. You can see in the visual it's all about nitrogen, baby. Remember, that was the first point I made. You want to know that amino acids have nitrogen, and that's big time because lipids and carbohydrates do not have nitrogen. So any question on the test that looks like this, check out this visual. That's going to be something huge on the test. They're going to want to point out nitrogen deficiency and excess. So any sort of nitrogen imbalance is going to have to do with amino acids. Just stop reading the question and go, boom, another question right for you, okay? Pay attention to that. As we continue to look at the amino acids together, look at tryptophan on that list. You see that? Do you guys know what that's a precursor for? That's going to be serotonin, okay? So make a note of that to your right. Tryptophan is the precursor for serotonin. And you also want to know histidine, okay? And let me show you a way this was asked on a previous examination. And you're going to hear me talk about this in the histology video as well, but you want to know histamine is a vasodilator. That's a big deal. It vasodilates, obviously, your arteries, right? But it's going to constrict your lungs. So I want you to say that to yourself. Histamine, vasodilator, all right? And the lungs, bronchoconstriction. That's a key thing you want to know about histamine. You guys can see as we read on in our notes, the paragraph that says multiple amino acids are linked together by a peptide bond, right? It goes on to say that's a covalent bond. And it gives some examples of that: dipeptide, tripeptide, polypeptide. You guys know the difference between amino acids and a protein is when you link 100 or more amino acids together, then you have a protein, okay? And that's going to be in the box right there in your notes. Take a look at that. A greater than 100 amino acids, now you have a protein, okay? So you guys can look here at the different functions of amino acids in the body. Look at that paragraph with me. It talks about the precursors, that some of the ones we've talked about. There's tryptophan right there, the third dash down, a precursor for serotonin. Tyrosine, a precursor for the DEN, like we talked about, DEN. Dopamine, epinephrine, and norepinephrine. Those are big points to know, okay? And even glutamate there. Glutamate being a precursor for GABA, all right? So hone in on that. Those are the key things I would pay attention to as you look at that, all right? Now, guys, take a look at that box with me where it talks about albinism. Now, put that visual back up here. Let's take a look at that again. You remember how tyrosine can be converted to DEN, D-E-N, but it can also be converted to melanin. You really want to know that enzyme of tyrosine to melanin, and it's tyrosinase, right? And this is the way this was asked on a previous exam. And it's not only been asked once, but several times. So you want to know this link really, really well. Okay guys, now we're going to talk about structural levels of proteins. Doesn't that heading get you just fired up? You guys remember there's 4 different types of levels, right? And the first one is going to be the primary level. And as it says here, this is the amino acid sequence, right? It's just, think of it like an old telephone cord, right? None of us have those anymore, but you can imagine these little cords right next to each other, right? Take a look at this visual just to kind of help you understand what I'm saying. What the primary level is just referring to the order of the rings. So you can imagine if the order was glycine, arginine, leucine, that's going to be different than, like, say, let's tyrosine, glutamine, and aspartate. It's just all about the order of the amino acids. And the way this has been asked on the old test is to know that this order that we're looking at determines the function of the protein, okay? So that's the way they can ask this primary structure. Now, I want you to also know that they've asked kind of the same 3 or 4 questions over and over on the structural level of proteins, and that's one of them to watch out for. Now, the secondary, as it says here in your notes, refers to the local interactions between neighboring amino acids. So go back to that same visual in your mind of these little rings next to each other, and you can imagine how the way that these individual rings interact with each other, right, could be different between one ring and the other. And by the way, where it says local interactions, make an arrow right above that in your notes and put non-covalent, okay? They could get picky on that. Remember, non-covalent means not sharing of electrons. And you can see here the examples: alpha helices, beta sheets, random coils. They haven't really gotten picky in the past about knowing the difference between each. They've wanted you to know, though, that these are examples of secondary structure. Okay, that's something I would pay attention to. Okay, guys, now we're going to look at tertiary structure. And I want you to circle that T of tertiary and write 3-dimensional, okay? And what I want you to see with this is a 3-dimensional shape after you've accounted for the primary and secondary structure. So going back to that telephone cord example, take a look at this on the visual. You can imagine, depending on how those local interactions are formed, it can give you a 3-dimensional shape that differs depending on one protein from the other. So it's just a 3-dimensional shape. Now, there's 2 examples of this: globular and fibrous. Globular, as it says there, is water soluble. Think of it like a glob that disintegrates in water, okay? Globular is water soluble. It says there that albumin is a classic example of that. Whereas if you go to fibrous, it's water insoluble. You can see there bolded collagen is your classic example, which anytime you see collagen in the notes, baby, you want to pause and destroy that. Collagen, we're going to get to in just a minute, is always going to be glycine plus an X and a Y amino acid, right? We'll get into that in just a minute. Now, this was a really hard question asked on old test about globular, and I want you to take a peek at that, and you can add that to your notes. Really, really hard, only asked one time, but in case they ask it, I want you to be all over it. Last but not least, here's one other question they asked about the structural levels of proteins that I wanted you to take a peek at. Another all-star question. When you get this right on boards, if they ask, you can walk out and be like, I'm the man or I'm the woman. You'll feel good about getting this right. Hard question. Okay, guys, now we're going to look at different types of proteins, and the first one is going to be enzymes. You guys can see as we glance at our notes together that enzymes lower the activation energy needed to initiate a reaction. You guys know that substrates bind to enzymes. I'll show you that in a second, an example of that and how they could ask you on the test. It'll come in just a minute. But I want to look first here at antibodies. You guys know antibodies are made from plasma cells, okay? Remember, plasma cells are going to be mature B lymphocytes. We've hit that over and over. You're going to see that again in histology, okay? And if we look at these different antibodies, you know that they spell out GAMED, okay? So as you think about that, there are 5 major types that we want to take a peek at. The first one is going to be IgM. And I want you to say this with me, all right? IgM is made first. So if they ask you if a patient is sick, like the primary exposure, the first time someone's exposed to an antigen, this is going to be made first, okay? Primary exposure. Now, if you go to IgG, I want you to know, as it says here, the most plentiful antibody. It's the only one that can cross the placenta, okay? The other thing to do with G that I would pay attention to is gingiva, okay? Like gingivitis, you want to think this is going to be the predominant antibody found in that situation, okay? That's been asked multiple times on old test. Pay attention to that as well. And you can take a look at the screen here to see an example of that. Let's go on to IgA next. You guys know it's the primary antibody in secretions, as it says there in mucus, saliva, tears, breast milk. Those are kind of classic things you've heard over and over. But the thing that I've seen over and over asked on old tests is when you think of IgA, think aggregation. Because what it does is causes bacteria to aggregate in the oral cavity. Okay, so this is a way it's been asked on old tests. Take a look at this to make sure you have this down. IgA, think aggregation. The other thing you'll see a lot in terms of test questions with IgA is knowing that it causes pathogens to not be able to adhere to mucosal surfaces. So aggregation and adherence are what I would pay attention to for IgA. See all those A's go together. Pay attention to that when you're studying. IgD, I always think of it like we don't really know. IgD, don't know. Not a real prominent function there. Not seeing any test questions on that. IgE though is huge. I mean, so huge, big deal. You guys know in systemic disease, when you review that chapter, you'll see we have hypersensitivity reactions. There's 4 types, type 1, type 2, type 3, and type 4. And type 1 should be where a lot of the test questions come from. You're gonna need to know that IgE is the prominent antibody in that situation. Okay, so let me take you through a scenario of this. You guys might remember the whole reason this is important, as it says here in your notes. It says IgE binds to allergen and triggers histamine release. As soon as you see that, you're thinking what? Vasodilation, yes. But let me kind of take you back to the basics. Look at this visual with me now, okay? You're gonna see this again in systemic disease. Make sure you remember though, that histamine is always found within mast cells, and what's the other type of cell? Basophils, yes, right there, okay? And the very first time you're exposed to an antigen, there's no symptoms, but something does happen, which is that IgE antibodies are produced, and they come and bind to the surface of this mast cell or basophil, right? Now, the second time an antigen comes into the picture, or another way to think of it is when the antigen is reintroduced, right, it will bind to that antibody. And by the way, that portion of the antibody you'll see in immunology that it's binding to is going to be the FAB portion of the antibody, right? When it binds there, it causes a conformational change in the antibody. And now what happens is calcium can enter the cell, right? When calcium enters, that causes depolarization, and now histamine is released. So I hope that really helps clear it up, but the bottom line is they could ask a type 1 hypersensitivity reaction so many different ways. So whether you nail it right now, this moment, if this is your moment for type 1, or if it's in systemic disease, whenever it is, absolutely obliterate this, because they could simply ask when questions like in a type 1 hypersensitivity reaction, what symptoms happen on first exposure? And your answer would be no symptoms, right? They could ask what happens though in terms of what happens in the body upon first exposure, and you would say IgE antibodies are produced and they bind to basophils and mast cells, right? I think the most likely question is to know that calcium depolarization, okay, is what leads to the histamine release. So dominate that. You're gonna see we talk a lot about histamine and wanting you to know that so badly because it's very likely to be asked. And so knowing how it gets released is critical. Okay guys, now we're going to talk about collagen. This is big time important, okay? Multiple test questions on this, and it's all right here in the notes. I want to point out the key features of what you should know. If you look at collagen, first line it says fibrous protein. That means water what? Insoluble or soluble? Water insoluble, yes. Next line it says you have 3 polypeptide strands. Pay attention to this number 3. Next line, triple helix. Yes. And next line, 3 amino acids, glycine, proline, that's a big one, and lysine. Okay, say to yourself, proline, that's a big one. And the reason why you're gonna see on the next page. But what I want you to hone in on now is what happens to that proline and lysine in the second and third positions. Take a look at this visual here to point this out. Okay, so here you see the structure of collagen. You see there's proline and lysine. And what's going to happen is vitamin C is going to come and cause hydroxylation of that proline and the lysine. Okay, now if you take a second and look back to your notes with me, you're going to see in the box, it's not just vitamin C that does that. It's also ferrous ions and alpha ketoglutarate. Okay, that's really picky, but I would know that those 3 things are what cause hydroxylation of those proline and lysine residues. Okay, that has been on old tests, so hone in on that. Every single one of you guys know that vitamin C is needed for collagen because you know without vitamin C you get scurvy, right? Yes, so the test question is going to be more likely to be what does vitamin C do, right? Why is it important? And that's the proline lysine residues, okay? Speaking of scurvy, look at that next paragraph where scurvy is bolded there. It says scurvy is caused by vitamin C deficiency, right? You guys see that? You guys know it can cause weakened connective tissue. It can cause problems with bleeding of the gums. That's a big one to pay attention to. Poor wound healing, etc. Okay? You guys also know here lupus and rheumatoid arthritis. These are going to be problems with collagen disorders, right? In particular, what I would pay attention to with lupus, do you remember the major sign on the face with lupus? Write it in your notes right there. It's going to be the malar rash. And just in general, you probably remember that lupus, it attacks multiple spots throughout the body, because if there's one thing I would know about lupus is that it attacks DNA. And you know you have DNA throughout your whole body, so make sure you kind of remember lupus, young females, attacks the DNA, malar rash. Okay, if you look at osteogenesis imperfecta and Ehlers-Danlos syndrome here in your notes, both of them are similar in that they affect type 1 collagen. Okay, what's the structure of collagen again? Glycine. What's next? Proline. Yes, lysine. Now, the proline I said, remember, was a huge deal. And look at the box here for a second, because this has been on multiple old tests. It says right here, the concentration of hydroxyproline, boom, there it is, may be used to estimate the amount of collagen within a given tissue, because that is a unique substance found in collagen. In fact, if you look at the next paragraph and compare collagen to elastin, you see a whole couple sentences there. Don't read it. The take-home message is you don't have hydroxyproline in elastin, okay? That's an important distinction you want to know. Okay, guys, now let's take a look at hemoglobin. Look down in your notes here with me for a minute. It says that hemoglobin transports oxygen and carbon dioxide throughout the body. You guys know it's a component of red blood cells. Now, it says in the next bullet point, there's 4 polypeptide chains, each have an iron-containing heme group that binds to oxygen. And what I want you to pay attention to here, take a look at that visual so you can see that, right? And the key thing you'll see in a second is that oxygen is bound to each one of these, right? If carbon dioxide gets bound to one of them, remember, the key thing to know is that carbon monoxide binds irreversibly, okay? That's a key distinction you want to know. The other thing is that iron in this situation must be in a ferrous or reduced state. And you can see that in one of the bullet points right above the box in your notes. It talks about iron must be in a ferrous or reduced state to do that binding, and that carbon monoxide, it says, binds irreversibly, right, leading to carbon monoxide poisoning, okay? Okay, guys, now let's look at enamel proteins together. I want to point your eyes down to where it says amylogenins in bold there, okay? If you read that, it says they comprise 90% of enamel proteins and are involved in the organization of enamel rods during tooth development. Everybody see that? So they're a big portion of enamel proteins. The question that we've seen a lot is in the box. It talks about this disease, amylogenesis imperfecta. It results in a loss of amylogenin formation, which means you're going to get a loss of enamel, right? So in that box, it says you get a thin hypoplastic enamel layer. That's a big deal. So that's been asked on past tests, and I want you to put an arrow to your right because there's a vitamin insufficiency that will also cause a hypoplastic enamel layer. So I want to group all that in one spot in your notes just so you can kind of go, whew. Okay, if they ask me anything about hypoplastic enamel layer, I got it, right? Anybody know what those vitamin deficiencies are? Because there's 2 of them. It's vitamin A and vitamin D. Yes, good. And if you didn't know that, don't worry. I'll show you that again in your notes later on in this chapter at the last portion when we talk about nutrition. Okay, guys, one more point about enamel that I want to point out that's in your notes. If you keep reading on, you're going to see enamel absorbs something. Anybody remember what it is? It absorbs fluoride, yes. And when enamel absorbs fluoride, you get fluoroapatite, right? So pay attention to that. That could be something it is something that's been mentioned on old tests and definitely could be on yours. So that's the last thing I want to hit on enamel to make sure you're rock solid for that. Okay, guys, now we're going to talk about the Michaelis-Menten equation. All right, this is fun. I know it's hard for a lot of us, and it was hard for me at one point in time, but I think we can really simplify it, and I can show you pretty quickly where they've asked this on old tests, okay? So what I want to do is start off with an example, okay? I want you to think of an apple in front of you right now, and I want that apple to be an enzyme, okay? And I want you to take a bite of the apple, and where you took a bite out of that apple, that is the active site. That's your active site, okay? And so your hand right here is going to be the substrate, and all day long you bind to your active site. That is your active site. You bind to it. It doesn't take much of you, just a little bit of you, and binds to it, and the reaction takes off and goes all the way up to the ceiling, okay? Think of the ceiling as like the Vmax, the highest you can get it, right? So that's a typical scenario of what happens. Substrate binds just a little bit, the reaction goes off the charts, all right? And so what I want to do is show you that visually right now, okay? So as you look at this visual representation of this, you see a scenario where just look at the x-axis, that's substrate concentration. Just a little bit of substrate causes this to go all the way up to Vmax. You see that? Okay, now watch what happens if they ask you on boards about a competitive inhibitor. So a competitive inhibitor is going to bind to that same active site that you've enjoyed your whole life, right? It's going to bind there. It's going to compete with you, right? And the key thing you want to know is anytime you have a competitor, it's going to decrease binding affinity. You're not going to bind with as much affinity as you did before. So if your binding affinity is down, okay, this is just a relationship you need to memorize. Binding affinity goes down, Km goes up. What do I mean by that? Well, that means, look at this graph, it's going to take more of you, more substrate concentration, right? You have a competitor now. So it takes more of you binding to this active site to overcome the inhibitor, but eventually you have the exact same Vmax. You see that on the graph? So the way they could ask this on the test is with Michaelis-Menten, competitive inhibition causes a shift in the graph to the right. You see that? Great. Now let's introduce a non-competitive inhibition. In this scenario, you have a competitor, but it's not binding to the active site, okay? It's binding to another spot on the apple, okay? And when it does that, the key thing you want to understand is that it does not affect binding affinity because there's no competition. So look at this on the graph. You can see it now starting to take shape, but eventually what happens is the Vmax ends up at a point that wasn't as high as it should have been, okay? So even though there's no competition, it influenced the overall velocity of the reaction, okay? So on the test, if they ask you about Michaelis-Menten with non-competitive inhibition, it's going to shift the graph down, okay? That's the key thing you want to pay attention to, okay? So that's Michaelis-Menten. Now, guys, I want to show you with Lineweaver-Burke. And if you look at your notes, it talks about Lineweaver-Burke takes the log of this equation, and ultimately what it's going to do is it's going to cause a linear representation. So instead of having like what we saw with the curves, now you're going to see lines, okay? So let me show you this here on the graph. And same principles apply. Competitive inhibition, the key thing to know, again, competitive inhibition, you have a competitor, so it's going to influence binding affinity, which means it's going to influence Km, right? And the other thing you want to understand is that it's going to result in the same Vmax, right? So with a competitor, you overcome that competitor and eventually you have the same Vmax. Whereas with non-competitive inhibition, right, you end up having a shift downward of Vmax. Well, remember, look at the y-axis here. It's 1 over Vmax. So the bottom line is it's going to shift in a Lineweaver-Burke. It's going to shift with non-competitive inhibition. It's going to shift everything up, okay? So let me show that to you in your notes. Guys, as you look at the summary here, you're going to see underneath competitive inhibition, a look-alike molecule mimics the substrate and binds to the active site. And it goes on to say in the bullet point, Km increases, right, due to decreased affinity, right? So anything I want to point out as we look at non-competitive inhibition is in bold, it talks about first bullet point, Km will be unchanged. But next bullet point, the Vmax will decrease due to inhibition of the enzyme. Last bullet point says Lineweaver-Burke will cause an upward movement as Vmax decreases, okay? So that's the way they would show that on the test. Last but not least, notice how on the Lineweaver-Burke, the competitive inhibition crosses the uninhibited line. You see that? So the phrase I would remember, just a simple takeaway, is say to yourself, competitive crosses, competitive crosses. That's a good way they can show that to you on the test.